All right. Okay. So, hello, dears, again. So now we're now at the second half na gyud of our lecture on biochemical testing for gram-positive cocci. And for the previous parts, diba, we have discussed the catalase positive. You have the staph and micrococcus, right? And mga different tests nila. So again, try to slowly internalize lang yun, hinahinay sa, like, okay, absorb sa nako muna ako mga tests for staph, muna yung mga tests for micrococcus, muna yung mga expected results sa kada test, muna ang mga reagents na nasa mga tests, alright? So, okay, now, alright, for this part, we'll now go to your catalase neg, okay? Catalase neg na mga gram-positive cocci in the form of your strep and enterococcus, alright? So, again, for classification, as you can see, we have the different species. And we classify them according to the presence of the Lancefield antigen, the hemolytic patterns, and their common terms. Of course, first is your Lancefield antigen was discovered by, again, Rebecca, okay? Rebecca Lancefield, all right? Rebecca Lancefield, and this is a C carbohydrate in the cell wall of these organisms, all right? So we have S. pyogenes, Strep pyogenes, of course, your flesh-eating bacteria. Yes naman, all right? Yung Lancefield antigen is, of course, your A. That's why it's considered to be your group A, Streptococcus. Hemolysis niya is, of course, a well-known beta-hemolytic. All right? Again, when you say beta, complete hemolysis or complete clearing around the colonies, right? Night clearing. Okay? Ang ginalyze ang RBCs sa BAP. That's why it's beta-hemolytic. And, of course, group A strep because, again, iyang Lancefield antigen is A. For S. agalactiae, Streptococcus agalactiae, that's your B, ang Lancefield antigen. Still beta-hemolytic. That's why it's known as your group B strep. You have S. dysgalactiae, Equisimilis, and Zoo epidemicus. That's your C, Lancefield antigen. Beta-hemolytic pa rin. That's why it's your group C strep. You have your S. bovis and S. equinus group. D. Lancefield antigen, alpha hemolytic or pwedeng wala. Non-enterococcus, viridans na strep. Okay? Alright? You have, the, of course, Enterococcus fecalis, Enterococcus faecium, E. durans, and E. avium. Lancefield D. gyapon, hemolysis types niya is alpha, beta, or none. And that is known as your Enterococcus na mga species. Alright. Next, we also have, of course, kanina mga na-asterisks na yung mga ubang isolates na dili hemolytic. Okay? But again, most of them, they are exhibiting this type of hemolysis. All right? Okay. Next, we have, of course, your strep pneumoniae. You have your pneumococcus, lancet-shaped. Lancet-shaped diplococci. Wag namang isa. That's your strep pneumoniae. All right. Okay. And very interesting characteristic po niya is wala siya Lancefield antigen. Hemolysis type, well-known alpha hemolytic. Okay. So, incomplete hemolysis na ay greenish discoloration sa imuhang media, right? Okay, and common terms is, of course, your pneumococcus. All right, okay. Na-record. Okay, again, so your pneumococcus, okay? All right, S. pneumoniae. And of course, last, S. anginosus, mutans, metis, salivarius, sanguis, ACFG, and daghan kayo siyang Lancefield antigens or pwedeng wala. Again, different types of alpha hemolysis po, hemolysis patterns. You have alpha, beta, or none. And this is known as your viridans na mga strep. So viridans kato na mga, like mga other, so when you say viridans, again, viridans, the murag different or mga less mga well-known na strep species. And most of them, if I'm not mistaken, they cause endocarditis. Because again, they're usually found in your mouth. Okay, so kana mga tooth decay, mga tooth na mga extraction. And if this bacteria is causing the decay, alright, and pwede siyang transfer sa imong bloodstream, abot sa heart, that's why makakause siyang endocarditis. Alright, but in a nutshell, muna siya mga different strep nato na species and their Lancefield antigen. Please take note, kinsa ganitong walay Lancefield antigen, you have strep pneumoniae. Alright, okay. Alright, so again, balik ta, gram stain, di ba? When we perform gram stain, we discover na it's gram-positive cocci. The first test that we perform is your catalase test. And of course, if catalase positive, you have staph or micrococcus. And if catalase neg, you have strep and enterococcus. And this time, what we're going to focus is the catalase neg na mga gram-positive cocci. Your Streptococcus and Enterococcus. All right. So for the first test that pwede natong ma-perform, for the rapid presumptive identification of catalase neg na gram-positive cocci is known as your leucine aminopeptidase test or LAP test. Dili siya WAP ha, dili siya WAP. Dili siya ang wet okay? It's LAP, leucine aminopeptidase test. By the name itself, it detects again the presence of the enzyme leucine aminopeptidase. Alright? And ang principle, leucine beta-naphthylamide, that's the reagent, in the presence of LAP, okay, it's now converted to beta-naphthylamine, and add na itong reagent, DMACA or your P-dimethylaminocinnamaldehyde. Ang resulting color is red color. Okay. Alright, so here's an example. Again, positive, red, and then negative kay yellow or walay color change. Alright. And ano presumptive ID siya? Because strep, your strep, okay, Enterococcus, and Pediococcus. Alright? These are all LAP positive. Okay? Alright. And the LAP negative are your Enterococcus and Leuconostoc. Okay. So these are all, they are all of the same family. They're under the Streptococcus na family. Alright? Pero again, rapid presumptive ID. Because again, pag positive sa LAP, pwedeng maka-think na ka na, okay, it could be Strep, Entero, or Pediococcus. And if it's negative, then it could be Enterococcus or Leuconostoc. Okay? Alright, that's according to Mahon. Alright? Okay, that's for LAP. Okay. After that, of course, we base now the tests for Strep and Entero based on their different hemolytic patterns. All right. So we again base it on the hemolytic pattern on sheep's blood agar. So again, we have three types, diba? You have beta, you have alpha, and gamma. All right. So again, these are example of the mga pictures sa beta, gamma, and alpha. As you can see, beta na clearing. Clearing, right? So complete hemolysis. Alpha, partial, na greenish discoloration, ha? Please take note, greenish. And gamma hemolysis, wala. The medium remains color red, pero naaray ni tubo. Okay, so meaning wala nila na-lyse ang RBCs. Alright, so no hemolysis or your gamma hemolysis. So we'll start first with your beta hemolytic na mga, or beta hemolysis, or complete hemolysis na mga reactions sa BAP. So what are the tests to perform after is your, of course, the PYR hydrolysis test and bacitracin susceptibility. Diba? Na-mention na ni bacitracin ganina sa staff. Of course, we use them again na pod for strep. And usually, mas definitive ang bacitracin for the strep species. Because again, remember, bacitracin is known as your taxo A. Ayan. Ngano siyang taxo A? Because it's usually used for the identification of the group A strep. Ayan, di ba? So, wag na mag-isip. Nganong taxo A, it's for group A strep. Di ba? Easy because your group A strep is bacitracin sensitive and PYR positive. Di ba? So, usually, ang characteristics ay mong strep payo, PYR positive, L-pyrrolidonyl aryl amidase test positive, and sensitive siya sa bacitracin. Ngano ganin siyang taxo A? Because it's usually ginagamit for group A na strep. And unsay reaction ni strep payo sa bacitracin, it's sensitive. All right. And kung PYR neg ka and resistant, we now subject it the next tests, Hippurate hydrolysis and CAMP test. Remember guys, 'di ba? Remember mo ganina sa table, kinsa tong mga expected na strep species na beta-hemolytic yun, 'di ba? You have strep payo, strep agalactiae, at mga viridans, and group C and group D strep. Usually, of course, if beta-hemolytic, the first organism na itong pwedeng ma-think is strep payo. The second is strep agalactiae. That's why we are having these types of tests because they are really pointing out to those organisms. Okay? So, munang PYR bacitracin, atong unang gitest for strep payo, and next na pod, kaning duha ka test, it's for strep agalactiae. Okay? Alright. So, we'll now go to the different tests. So, our first test is your PYR hydrolysis. By the name itself, you detect the presence of an enzyme na pod, L-pyrrolidonyl aryl amidase. Now, it's quite the same principle with your LAP test. Medyo naari different sa reagent. Ang reagent na gagamit is, this time for PYR is L-pyrrolidonyl alpha naphthylamide. And in the presence of your enzyme, may mong alpha naphthylamine. Same reagent yapon sa leucine, di ba? LAP. DMACA or P-dimethylaminocinnamaldehyde, and still the same reaction na ay red color ang positive. So example, here's a picture. Of course, the left is positive, right? bright red color within 5 minutes, and negative is of course walay color or orange ang color. Okay, so again, that's for PYR hydrolysis. Basta gani PYR positive, kisa to PYR positive, you have strep bio. Alright. Okay. Alright, so muna siya for PYR. Diba? Remember, if PYR neg, niya resistant pag ito bacitracin, we now perform Hippurate hydrolysis and CAMP. Because, why? Because we are assuming now na dili siya S bio, but it could be strep agalactiae. Because again, it's still beta-hemolytic. Remember? So again, strep agalactiae, Hippurate positive, CAMP positive. Yeah, if it's negative or CAMP neg, it could be, okay, we now subject it to the different tests na pod, which is bile esculin. And if bile esculin positive, then it could be, okay, if bile esculin negative, it could be the other beta-hemolytic strep na dili group A, B, or D. And if it's bile esculin positive na pod, ato na siyang isubject to PYR hydrolysis and 6.5% NaCl. So again, that's the parang flow chart, flow niya. Alright? We'll discuss this sa later part. Okay? Alright. That's for the Enterococcus na mga species o viridans. Alright? Okay. Alright. So first is of course Hippurate hydrolysis. Still the same, we detect the presence of Hippuricase. And our reagent is ninhydrine. Ayan. Ang principal ani guys is Hippurate. In the presence of Hippuricase, mahimo is may mo siyang sodium benzoate and glycine. Now, glycine will react with ninhydrine to form NH3 and hydrin dantine and ninhydrine residual. And this complex or this products will create a deep purple complex. Okay? Alright. So, ayan. So, here's the positive, deep purple. Negative is absence of color or slight ra ang purple or yellow pink. Basta gani deep purple ang positive. So again, yung reagent is ninhydrine and detects the enzyme Hippuricase. Now again, this is kung positive ani for gram-positive cocci, it's your strep agalactiae. Now we also have a gram-neg bacilli na positive ani, and that is your Campylobacter jejuni or jejuni. Alright? Campylobacter jejuni is positive for Hippurate hydrolysis test. Pero it's a gram-neg bacilli. So, Sir Mark, unsa man na? Positive ang strep agalactiae, niya, positive po ang Campylobacter. Di na ako malibog ana? Of course not, chok not. Because, remember, this is gram-neg bacilli. So, pag gram-stain pa lang ni mo daan, makita na ni mo na bacilli siya. And your strep is, of course, strep agalactiae is cocci. So, di mag-guol, di ma-shocked. Okay? Alright. Next, you have your CAMP test. Okay? CAMP test, unsay meaning sa CAMP? Again, it detects the CAMP factor or your Christie-Atkins-Munch-Peterson factor. Okay. Now, again, ang principle ani niya, your Staphylococcus releases a beta-lysine. So, when you say beta-lysine, so it lyses the blood agar, sa blood agar plate. And in the presence of the CAMP factor, they will work synergistically, meaning ilang mag-work sila together. Therefore, muna ang positive result mo is enhanced hemolysis or arrowhead hemolysis. Ayan. Basta gani, sa boards man good, nailo ni gawas. What is the positive result of your CAMP test? Niya, wala may arrowhead. So therefore, enhanced hemolysis ang answer. Okay? Enhanced hemolysis or arrowhead hemolysis. So here, as you can see, so this is the Staph aureus, guys, of course. We need a Staph aureus. Gi-inoculate sa daan. So, in a plate, okay? So, you inoculate first Staph aureus. And then, so this is Staph aureus. And then, you inoculate the test organism perpendicular. Okay? Perpendicular to the S aureus. Alright? And if mo create sila og, again, arrowhead, bow tie shape, or again, enhanced hemolysis, then it's CAMP positive. Alright. Okay. So, arrowhead hemolysis or enhanced. And negative, when say negative, no enhanced hemolysis. No enhanced hemolysis. Okay, muna siya negative. Alright. Ayan. So, again, kinsay positive sa CAMP test, you have strep agalactiae. Now, for the gram-positive bacilli, ayan, for gram-positive bacilli na po, we have a gram-positive bacilli na positive sa CAMP. Basta na-mention ni Ma'am Teddy. Kinsa man to? Si Listeria monocytogenes. Yes naman. Listeria monocytogenes is CAMP positive. Okay? So, napod siya ay CAMP factor. So, please take note. So, still the same scenario with Hippurate hydrolysis. Sir Mark, oh my gosh, positive ang strep agalactiae, positive po ang Listeria monocytogenes. Paano na to? Of course, Again, sa gram-stain palang daan, you will see na gram-positive bacilli ni, bacilli ang shape, and of course, Streptococcus is cocci. So, dito palang daan, di na ka ma-worry. Okay? Alright, ayan. Listeria monocytogenes. So, sa kanyang characteristic motility, sige, nagbalik-balik, you have tumbling, the tumbling motility. Alright. Okay, so again, that's for the CAMP test. Okay, next you have bile esculin hydrolysis. By the name itself, it determines the ability to hydrolyze or to process esculin. Okay? And esculin, I think, I was searching on such a type. I think it's a type of sugar, mantingali. Or parang, yeah, it's a type of sugar, I think, if I'm not mistaken. Okay, esculin. Inhibitor niya is 4% bile salts, of course, to inhibit other organisms that could be present in the sample. Para ang mutubo ra gyud is the strep species that you want. Okay? And the principle in the presence, esculin, in the presence of hydrolysis, ang result niya is glucose and esculetin. Now, esculetin cannot be visualized, okay, in the media. That is why we add a ferric ammonium citrate because mo combine ang ferric ammonium citrate and esculetin to create a black precipitate. Okay, so if wala ferric ammonium citrate, dili ni siya ma-detect. So the medium stays as it is. So dili ka makaingon if positive or negative. But in the presence of ferric ammonium citrate, muad siya sa esculetin, okay, and a black precipitate is now formed. Okay, that is why makaingon ka na, positive siya for bile esculin hydrolysis. Okay? Alright. So here's an example again, na black precipitate. That is because of ferric ammonium citrate and esculetin, so meaning positive, na hydrolyze niyang esculin to esculetin, okay, so positive, and negative if no color change. Alright? Ang positive sa bile esculin hydrolysis is blackening. Alright, okay. Ayan. Okay. Alright, so again, di ba remember, if bile esculin positive siya, we subject it to PYR hydrolysis and 6.5% NaCl. Because again, if it is PYR positive and 6.5% NaCl positive, it's your Enterococcus species, okay? And if it's negative, it's your Streptococcus na group D. Because again, remember, Enterococcus and group D strep, they can be beta-hemolytic also. That is why naa siya diri, under beta-hemolytic, okay? Alright. So your salt tolerance test or your, again, 6.5% NaCl, quite the same rationale. You want to determine if the organism can tolerate or can withstand high salt concentration. In this case, 6.5% NaCl. Alright, so still the same procedure. You inoculate the suspect organism on BHIB or brain-heart infusion broth. So this is again a broth, brain-heart infusion broth na naay 6.5% NaCl. And incubate, if mutubo, then therefore makatolerate sila sa 6.5% NaCl. So you observe for turbidity. Alright, so here's an example. So the left is positive na may turbidity, meaning nakatubo ra sila. They tolerated the 6.5% NaCl, and negative, walay tubo because dili nila kaya ang 6.5% NaCl, therefore walay tubo, absence of growth, clear suspension, a clear solution, okay? All right. Ayan. Okay. And we also have what we call your 20% dextrose strip test. So your purpose lang is for subgrouping beta-hemolytic strep. Kaya dagan man kayong damay ubang daghang beta-hemolytic strep, di ba? You have group A, B, or C. So ang procedure ana is you place a 20% dextrose strip at the center of a BAP and then inoculate 2 stabs, one dool sa strip and one away from the strip, and then you observe for hemolysis. So here's an example. This is the 20% strip. You inoculate, okay, stab, near, and far. Oh, yeah, wherever you are. My heart will go on, char. Again, first stab near the strip and second stab is away. All right, okay. So we have the different results. Decrease in size of hemolytic zone. So, meaning, mas dako ang zone kaysa diri, niya magka-alpha hemolytic pa duol, then that is characteristic of your group A strep. Okay? Next, if increase in size, so mas dako ang size compared sa layo, alright? And with or without brown pigmentation, then that is your group B na strep. So, as you can see, mas nagkadagko as nagkaduol siya sa dextrose strip. Okay? So, therefore, it's group B. And no change in the size of hemolytic pattern or no change in hemolytic pattern, that's your group C na strep. So this is based on the ability of the organism yata to tolerate the 20% dextrose, okay, if ganahan ba siya or dili. Alright? So again, for group A, magkagamay ang hemolytic pattern or magka-alpha hemolytic siya as nagkaduol sa strip, group A. Group B, nagkadako ang hemolysis and walay, with or without brown pigmentation as nagkaduol sa dextrose strip, then that's group B. And no change in size, it's your group C na strep. Okay. All right. Okay. All right. So for the next video, we'll now go to the alpha hemolysis ng mga organisms and the different tests to perform. Okay? All right.